Good morning. Today's class will be discussing about the transcriptional regulation of plant genome. So before going to the topic, we should know what is transcription. Transcription is a process by which the information in a strand of DNA is copied into a new molecule of messenger RNA, that is mRNA. Or it is otherwise called a process by which the information stored in the DNA is transferred to an mRNA through the synthesis of RNA is known as transcription. So coming to the transcription and the central dogma of life. So transcription, we all know that, which is the fundamental process that takes place in our genome, and it is the process of converting turning the DNA into an RNA or synthesis of RNA. Okay, and coming to the central dogma of life, so which will gives an idea about how the genetic information, that is the flow of genetic information from DNA to RNA, and thereby which will makes the functional product, that is protein. So what are all the components that are needed for the transcriptions are transcription? So it is in need of a gene. So gene which acts as the functional unit of DNA that can be transcribed, and RNA. So this RNA can be synthesized from the DNA template that catalyzed by the RNA polymerase enzyme, and template strand of DNA. So template strand of DNA which contains one of two DNA strands, such as non-coding strand, and the other one is non-coding strand, that is antisense strand, and the other one that is sense strand. Okay, so which will produce more copies of RNA molecules, and this sense strand, it is called as a non-template strand, and it doesn't participate in the transcription. Okay, so which is otherwise called as coding strand of the gene. Okay, so next one, that is it needs some primary mRNA such as codons for same base sequence as coding strand, and some of the substrates, so which may be of ATP, GTP, CTP, and UTP. So which may be of ribonucleoside triphosphates, and enzymes such as DNA dependent RNA polymerase, that is very much responsible for the synthesis of RNA in 5 prime into 3 prime direction. Okay, so with the help of DNA template. And what are all the characteristics of transcriptions are? So this transcription is very selective, and here the entire molecule is not expressed in transcription. Okay, so that is that's why the transcription is very selective. And the second characteristic, that is RNA are synthesized for selected regions of DNA. Okay, so now we will move on to the next. So here this slide represents the difference between the transcription in prokaryotes and eukaryotes. We all know that the transcription in prokaryotes and eukaryotes are differ. Okay, so the basic difference between the prokaryote and eukaryote is, so in case of prokaryote, both the transcription and translation that takes place in cytoplasm, whereas in eukaryotes, the transcription that takes place only in the nucleus, and the translation that takes place in the cytoplasm. So that is um, both prokaryote and eukaryote, the transcription and translation that takes place in different compartments. Okay, and the second difference, that is um, in prokaryote, both the transcription and translation occur simultaneously, but in case of eukaryotes, it doesn't occur simultaneously. Okay, And another difference, that is RNAs are released and processed in the cytoplasm in case of prokaryotes, and coming to eukaryotes, the RNAs are released and processed in the nucleus. Okay, and for the enzymes involved are here both the eukaryote and prokaryote involves RNA polymerase, but the number of RNA polymerase involved will be differ from each other. In case of prokaryote, only one RNA polymerase will be involved, whereas in case of eukaryotes, there are three RNA polymerases are being involved. Okay, and another difference, that is in prokaryotes, it doesn't need any factors or transcription factors or any other proteins for the initiation of transcription. And in eukaryote, it requires some proteins which are called as transcription factors, that is very much essential for the initiation. And in prokaryote, the processing of mRNA for the that mRNA will be of a polycystonic, that is which can uh, which will code for more than one polypeptide. Okay, and in eukaryote, one mRNA will code for only one polypeptide. And the another major difference between prokaryote and eukaryote is, in case of prokaryote, there is no processing of mRNA, and in case of eukaryote, there is a processing of mRNA is essential. Factor 2H, which will play an important role in promoter, in releasing the promoter. Okay, so these are all the significant roles of each transcription factors. And for the promoter release and open complex formation, so some of the hydrolysis of ATP is being takes place by the RNA polymerase, and this hydrolysis of ATP is mediated with the help of transcription factor 2H. And this transcription factor 2H is an 10 subunit protein, so which contains both ATPase and protein kinase. Okay, so which will form the transcription bubble, and finally the promoter will be escaped. And the next step, that is elongation. So elongation here, the once the promoter are escaped, and which will shed most of the transcription factors for initiation. So this polymerase enzyme occurs some of the uh, adapted to the new factors. Okay, so there the double stranded DNA that enters from the RNA polymerase mm, is unzipped to avoid the template stand for RNA synthesis. Okay, so that takes place in the elongation, and here the DNA RNA hybrid form base pair is being formed. And some of the elongation factors are needed, so which will stimulate the elongation process in the transcription. Okay, and some elongating polymerase along with some of the protein factors are being involved, and during the uh, elongation process, some of the mechanism that takes place. Okay, so such as polyadenylation, capping, and splicing. So we'll see that process in the upcoming slide. And the last one, that is a termination process. So here, once the RNA polymerase reach the terminator, which will give some signal to the RNA polymerase to stop or dissociate the complete transcript, and which will release the RNA polymerase from the template DNA. Okay, so this release of RNA polymerase from the template DNA will be vary or differs for each of the three RNA polymerases. And this termination process that takes place with the help of factor dependent or factor independent. And for the factor dependent, it needs some specific transcription termination factor, and for factor independent, it doesn't. So for the factor independent, the RNA polymerase 3 itself can terminate the transcription efficiently without the need of additional factors. Okay.